Hi everyone. Welcome for the week 4 of our intermediate course for the SynBio4All Africa. My name is Ice. Let me change my name a bit. Ice. Okay. Yeah, my name is Ice. I'm gonna be the instructor for today. Let me try one quick thing by sharing my screen. And if you see my screen okay, then that's perfect. If you guys have trouble seeing my screen, please let me know. Like you can type in the chat or like unmute and let me know. People are still joining the room right now, so like we'll be talking about some logistics or some tasks about our group project before we move forward. All right. For the sake of time, let's chat about the group project a bit. So, you probably heard from Heidi about our group project that is ongoing, right? And like in a few weeks you'll be starting working on this group project for real. So this is a summary from what Heidi mentioned last time, where we want you guys to form a team. It can be up to 5 people on a team. And we want you to focus on important social problem, and that problem should be able to solve with a synthetic biology solution. You don't have to do it for real. We are working on a proposal so that like we can think about what could be a good solution for these problems that we're seeing. So it would be great if you can form a group now within this week or next week, and then submit ideas on like what you want to work on week 7. If you're looking for some ideas to start working on like these relevant ideas, you can check the syllabus, as there will be some relevant information about like some potential ideas that you can work on it as well. And we will ask you to do 3 things, which is like present your ideas, respond to the question that people have, and also like provide feedbacks to the other groups who are presenting their projects. So week 8 to week 10 that we have scheduled will be dedicated all for the group project. So the other thing that I want to check briefly, I'm gonna launch a poll and see if you guys can see the poll. If you can see it, please like respond as you wanted to. Like there's nothing wrong. I just want to check whether you can hit onto the poll. Okay, I can see some responses. That sounds good. So I have like maybe 2 or 3 questions along the lecture that I want to ask you guys on like what do you think about these? So that would be good that you know how to use the poll option in Zoom as well. Okay, I think that's what I want to convey on behalf of the instructors. Is there any question about the group project? I can probably like respond to whatever question you may have right now before we move forward to the lecture in like 2 or 3 minutes. See some chat.
I have a question.
Yeah, please.
About the project, I would like to ask if we need to add the mentor, or if we can give us the mentors in our project, or if we will do it as an assignment. Thank you.
Can you say that again?
I said, I'm asking if we would provide the mentors in our project, or if we will do it as an assignment.
Yeah, so us instructors, we serve as your mentor. So in the last like week 8 to 10, we will work together. If there is anything that you wish us to be helpful on your like project working, then we'll try to help as much as we could. But again, like all of you will have us like the team of instructor who has been teaching in the past few weeks to help you on this final project. So it's fine to like reach out to us and like ask further question about the project. If you have other people outside of the class who are willing to help, that's completely fine. Like we are not against that, but you know, we just want to be able to help like as much as we could. So about the question, like I think Sei asked about like we were told that some project topics will be provided. Yes, you can take a look at the syllabus. Some project topics will be provided, but you don't have to choose exactly what was provided. You can change that a bit depending on your interest. But we encourage you to choose around those topics so that like it's a topic that we can help the most, you know. So like if you choose something that is close to those topics, then us instructor team know what those are, and we probably be able to help the most. But when we don't want to limit your creativity, of course. Like if you have anything that you feel strong about to work on, you know, please give it a try. All right. I think that's clear enough. So we will go ahead and move to our lecture for today. Again, thanks everyone for hopping on, and for those who are listening to the recording. This will be around like 45 minutes to 1 hour, depending on like the question we have. So let's get started. Again, the class today will be about metabolic engineering. So in the previous classes, we have talked about like some engineerings, starting from the protein engineering and also the strain engineering. So those two things are very important for us to work on the engineering of the bigger scale. So here we are talking about the metabolism of the whole bacteria or the whole microbes. It might be yeast, it might be other organisms. We are trying not to touch, you know, plant or mammalian cells because it's very hard. But we will walk you through how do we deal with these approaches. And then at the end of the day, we might be able to like, you know, get some ideas how to work with something that was more difficult, like plant or mammalian cells, like human cells. Okay. So the overview of today's lecture, we will talk about three main things. First, I will walk you through what exactly is metabolic engineering. So we will like go deeper into like what is the difference between metabolic engineering and the regular chemistry or chemical reactions. So we will compare that and like consider them as a factory where you want to use the factory to make something, right? If you think about factory, you may think about like something big, something that has like some chemicals in it, something that was trying to make some plastic bottle. Those like that kind of a factory. But in this case, we're gonna zoom in and take a look at our microbe and consider them as a microbial cell factory. So that's you know, that hopefully like, you know, encouraged you to think more. Why do we have to engineer these big metabolism systems? Second, we will go through like, you know, one angle of metabolic engineering, which is trying to discover new chemistries for the metabolic engineering. There are two key angles, which is like trying to look for new enzymes and also like trying to engineer and screen for new enzymes. And the third one is like the core of metabolic engineering, which is like trying to optimize whatever is available in the systems and try to make it work better. So think about like we have this factory already, but you want to make sure that people in the factory are working, can coordinate like between each other as good as possible. So that's what we are thinking about. Like we will find the right strategy to do it, and we will see if we can tune these chemical reactions, like multiple chemical reactions inside our system. Okay, let's get started. So what you're seeing right now on your left-hand side is the chemical plant for using like, you know, the old school. Actually, it's not that old. It's just like 100 or 200 years ago, but synthetic biology is much newer. So on the left-hand side, it was around like, you know, the plant that has started from like 100 years ago. That was the plants from BASF, the biggest chemical company in the world right now. And on the right-hand side is the plants that LanzaTech has collaborated with ArcelorMittal Steel. So that plant were the system that LanzaTech used the microbes to capture carbon dioxide and turn them into some other like materials. One key thing that I want to point out here is that like if you're using the chemical plant, you probably use petroleum or like some other crude oil as a starting material. But if you work on the microbial bioproduction, you're probably using something that was way more renewable, something like like something from plants, something from like animal feces, something like that. Or you can find something in the middle where it was like some chemical waste or like some recyclable stuff that can either go to the chemical plant or the bio plant. So briefly, if we go into the specific compounds that we want to make, this is the chemical called acetone. Acetone is like something that if you guys have used anything related to like nail polish or like something that was like trying to dissolve some color paint, acetone has been used a lot, and like acetone has been a very important chemical in our industry. So how do we make acetone? If we do it in the chemical way, then you have to take these benzene ring and these like propene with oxygen, put a lot of heat and pressure on it, and then you get acetone and phenol. This is the main process to get these compounds. But is it possible to make it using a biological systems? The answer is yes. There is this organisms called Clostridium acetobutylicum. Acetyl came from acetone, of course, and like from what you can see right here, it actually can take glucose and turn them to some other compounds that at the end it will turn into acetone in this pathway that we call it ABE pathway. And ABE stand for acetone, butanol, and ethanol pathway. And you will see these three compounds. Now I have a question for you. How many chemical reactions do you think are in this ABE pathway? Let me put another poll on. All right. I'll end the previous poll and launch a new poll. So, if you can see the poll, you can try to answer how many chemical reactions are there in this ABE pathway. All right. I see a lot of answers going for 10 to 20. I see some going for less than 10 and some going for more than 20. So while you guys are putting your answers in, anyone want to like jump in and tell us or share with us like how do you get the numbers? Like how do you know that it's around like 10 to 20, or how do you know that it's more than 20? Feel free to unmute yourself if you would like to.
Maybe we'll count the.
Hi. Sorry, like you guys were talking like together. Khadija?
May I say?
Yeah.
Yeah, myself, Khadija. Actually, I count them before you asking. So I found there is an 11 reactions in this pathway. So I thought it would be between 10 to 20.
Mm-hmm. Cool. So you actually count the numbers that was shown in this picture. Very good. And I believe like the other person probably has the same kind of answer. So yeah, the correct answer from this figure is like around 10 to 20. And you can see that like some R that was shown here were actually the rate of reaction. Let's say if you focus on, I hope you can see my cursor here. If you take a look at glucose turning into fructose, is R1, that's the rate of glucose turning onto fructose here. Or if you take a look at R15, which sort of suggests us that there are at least 15 reactions here, right? So like the first answer is likely not correct in this case. The reaction number 15 is when butyryl-CoA turned into butanol. So based on this figure, we have around 10 to 20. But in fact, there are much more chemical reactions inside the cell. So whenever you're trying to control this system, you have to deal with a lot more chemical reactions that is going on. All right. Then what about the chemical reactions? Like if you take like these, like, whatever 6-membered ring, we call it benzene here, and this propene and this oxygen, and turn into these two products. How many chemical reaction is occurring in the chemical plant? I think you know the quick answer. The answer is just one. There's only one chemical reaction occur in the chemical plant. Yes, that very good, Joy. Like that's a great answer. So the fact that like there's only one chemical reaction occur in the chemical plant means that like it's a lot easier to control, right? Compared to the bio plant where like there are a lot of chemical reaction occur when the cells are growing. That's why trying to engineer or control the metabolic system is very hard. So let's take a look at another system. Previously we talked about acetone, right? And like it's a lot easier to make acetone with a chemical process. What about ethanol fermentation? What you're seeing here is what we call baker's yeast. Yeast has been used for like brewing beer, brewing some, brewing some other alcohol. And so far, our like global society has been relying on these like ethanol fermentation a lot to make ethanol. So this is like, there was one time around like, 60 years ago that people were trying to use the chemical process to make ethanol. But it was used for only like less than 10 years. After that, like we stopped using that because like it doesn't worth it. It's a lot easier to make ethanol with the fermentation pathway. So hopefully it can convince you that like there are some compounds, there are some system that is much better to make through the biological pathway. In this case, like majority of biofuels nowadays are bioethanol, and they were made by this fermentation system. You just like throw corns, throw sugar cane in, and these yeast would just like keep eating those sugar and turn them into ethanol through that processes. Now let's take a step back and see like, if instead of like seeing the cells as factory, inside the factory, is there like something, there's some other like, chemical reactions that are very, very complicated. What I'm showing you here is one of the most complicated train system in the world in Japan. This is like the map of like railways in Greater Tokyo, like that means like beyond Tokyo. And if you zoom in, you still see something that was still very complex, right? So there's no there's no hidden agenda or like this map any I just want to show you that like these kind of maps seem to be very complicated, and it's actually resembling a lot of like unseen connections that is going on inside these like train maps. The same goes for this metabolic map. Some people even call them the metabolic metro map because like it resembles this kind of railway or train system. Like when you have the roads connecting together, there's a lot of like chemical pathway that was connecting to each other. And if you try to list all the chemical reactions that can occur inside the cell, this is a very complex and complicated chemical systems that's going on. Again, bottom line, I want to emphasize that like the metabolism that we are seeing here is very complex, and it requires a lot of work for us metabolic engineer to control them to make the particular chemical that we want. So what have we done so far? This is like not too recent, but like a pretty good review from Nature Catalysis, showing that like on the left-hand side, the compound that were highlighted were compound that can be made through biological system only to make those compounds, and they can actually feed into the chemical industry. So that means like, a lot of like biological systems are getting very important into our society these days. If you're losing some of these biological system to make these chemicals, then, the society, we lose a lot of like supply chains to make these chemicals. The other graph on the right-hand side is like some chemicals that were made through both biological system and chemical system. This means that like you can use biological system to make like a few steps earlier, and then use the chemical system to finish the whole thing. And that's also very valuable because like sometimes the final step or like the final two steps of biological conversion is just very hard to develop. And sometimes like chemistry is still easy enough for us to do, but our goal is to make everything bio-based. All right, now that we have these things in mind, we have two key challenges. If you want to make this system or this society much better, one thing that you want to care is you want to make more products available to our system, right? And the second thing is that you want to make these products, like this conversion, having more use. If you put 100% of like your sugar in, you want a lot of your desired product out, not just some random products. So we focus on these two things for the metabolic engineering lecture today. Now let's get into some of the chemistry that we're working on. I'm trying to simplify them, so hopefully like it's not complicated. It's not too complicated for you guys. So seeing on the left hand side here as like some metabolic network, if we simplify this network to like, let's say 3 or 4 steps of A turning into B, turning into C, and then D, assuming that like we start from something that was carotenoids. Carotenoids is like a group of compound that if you heard of like, natural rubbers, those natural rubbers came from this group of carotenoids compounds. If we make E. coli being able to make these carotenoids and put another gene in, and that gene was called CRTL, then they will be able to make the lycopene. If we add another gene in, they'll be able to make the beta carotene. And if we add just another gene in, you'll be able to make the zeaxanthin. These compounds has different colors, and they were corresponding to different pigment in these vegetables or like fruits, like these tomato, carrots, and also corn. So if you put them into bacteria, they will have different color. I see a hand raised, so please feel free to unmute yourself.
I just have a question.
Yeah.
So you said about how it's very difficult to engineer this and how complex it is. Then compared to like chemical reactions, then isn't that a disadvantage? Like we are going for complexifying the process, which means the processing will become more difficult and so on.
Yes. You're totally correct. Most of these metabolic engineering will be relatively difficult compared to the chemical process. But there was one important thing from the chemical, when we compare the chemical and the biological system, is that like the chemical system usually use the non-renewable sources. They were using like a lot of like petrol or like some like crude oil as a base, as a starting materials to make any chemicals. And we're going to run out of that at some point, right? Compared to using this biological system where you can just, like keep growing plants forever. And plant would just capture the energy from the sun. And this process, even though it's a lot harder, but they're not gonna end. We're not gonna run out of these plants. Does it make sense?
Yes, understood. Yeah.
Yep. So we are doing the hard thing because we have to. Thanks a lot for the question. That's a very good question to get started. All right. So with this system in mind, I have another question coming. So let's think about like if you're taking this CRT pathway from plants and put into E. coli. Do you think it will function right away, or it may not work? Let's see. I'll give you some time to think about it. So this is a good pausing point to think about like what you have learned in the previous lectures as well, right? In the first lecture, we learned about the overview of synthetic biology, what we have learned in the beginner class. The second class, we learned about protein engineering, and the third class, we learned about strain engineering. So when you think about the strain engineering, when you're trying to put the new genes in, the new foreign genes into the organism, you may have to consider putting not just the actual gene that is going to turn into the protein, but how to express the gene as well. So there'll be some promoters, there'll be some terminator or some ribosome binding sites that might be important for those construct. So thanks a lot for the response so far. The answer is it's hard to know. So if the gene cassette that you put into E. coli turns out to have the promoter to express a gene, then it will work. That means E. coli is smart, but that's not always happening. Sometimes E. coli doesn't know the language of the plant, and they may not express the gene properly. So for example, if they know what CRT expression look like, what the CRTL expression look like, then they will turn red. But if they don't know how to express CRTY to make this orange compound, then they will just stay red and not turn orange at all. And that's like, a pretty simple, understanding and concern for us. If we were to make the compound C or compound D, we have to make sure that all these genes were expressed properly. Otherwise, it would just, just sit there and not getting expressed. Think about like you have like some files in your phone, if you download the picture or video, or our class recording to your phone, but you don't really know how to open that file. That's something like that. You put into E. coli doesn't know how to use those genes. It will just stay there and not function at all. All right, let's try to make it a little bit more challenging. Let's think about trying to make compound X starting from compound D. And this compound X can be made by add another CRT gene. It's called CRTW. And if you have this CRTW, you can convert zeaxanthin, this compound that looks like kind of yellow, to astaxanthin that looks like pinkish red. This is a compound that we found in shrimp. So a lot of shrimp, or like like small crustaceans, like these small animals, they keep eating the red algae that we're making astaxanthin so that they turn red. And if you know flamingo that has like a pretty red, like legs and beaks, they eat these shrimps and then they turn themselves into the pinkish color because they keep accumulating this astaxanthin compound. So this is like, one example of like, if you're trying to make something to work, you have to put all the genes in, otherwise they're not going to work. This can also be expanded further. Let's say you're trying to make bacteria to make these compounds, but you don't really want to eat the bacteria, right? What if you put into plant? We are not touching that for this class, but this is for inspiration that like the picture shown, the picture shown here is the rice that was genetically engineered to make these compounds. So you can see that like they're making the yellowish compound and also the reddish compound. And this can be, this has been done, and like you can probably like try to introduce some of the pigments that usually have to eat a particular vegetables to get these nutrients. You can put into rice, where some people in the world, like some region in the world, were mainly consuming these rice, like as a core, like carbohydrate sources. So this is pretty interesting, right? Because this color of the rice are pretty obvious that they have these genes to make the products called carotenoid. However, like, what if we don't know the gene that we want to make? Like, for example, we want to make this product Y, and we want to make it blue, and we don't know. We don't know what enzyme we should be using to make these bacteria turn blue and make this blue pigment. And it's kind of hard, right? We don't really see a lot of blue stuff outside in the wild that much. Like if you're looking for like the blue flowers, that's pretty hard. That's relatively more difficult to find the flower that was white, something like that. So if you think about it, we may need new enzymes to make sure that we can do these new chemistry. And that's what we're going to dive into for the actual process. So this is a figure from one of the current opinion publication that I was part of. So you can see here that like to get new enzymes, there are multiple ways. You can either go through like the enzyme discovery pathway, where you're trying to look for the enzymes that are already existing in the nature, and you're trying to fish it and screen and look for the right one. Or you can take the enzymes that is already available, you know how it is, but you want them to perform differently from what it used to be. For example, they used to be able to turn, these like these yellow compound into the pink compound. You want them to turn the red compound into the blue compound instead. That was something that like, you might want to think about, and how to engineer these enzymes to behave accordingly. So there are several discovery aspects. You can take some samples from where you think it would be. Let's say you want to take something that might be able to degrade plastics. You may want to go to the place that you have, the plastic landfills, because like some of the microbes around there will try their best to eat plastic that was around in the landfills, right? And you might be able to find some enzymes that can degrade those plastics. You can compare those enzymes together and try to look for some scaffolds, like which is the keyword for like some part of a protein, and see if we can find some homology. Homology means some the similarity of those enzymes, and then see further if we can find new enzymes that might be more functional. Or we can take those enzymes and do what we call directed evolution, by forcing them to behave better in a certain situation, or like get the structure of them, see what modification based on our knowledge, our design, to see if we can make a better enzymes. we can try to compare with a lot of enzymes and see like what is the general trait by looking at the ancestral sequence and reconstruct them. Maybe that will help us like get some promiscuous activity. Promiscuous means something that they usually doesn't work on, but they can do it. Or the de novo protein design, where we just like start from scratch and try to design a completely new protein. So with these processes, I'm showing you one example from one of my colleagues. This is the enzymes that can convert this amino acid into this amino acid that has this oxygen group. So this enzyme has been known, right? But it's not easy to find something that can put oxygen onto these compound that doesn't have nitrogen. So this is the goal of this particular screening. They take this compound that has like very similar structure to this amino acid right here and try to do this, try to do this hydroxylation, like putting this OH on using the same enzymes. By looking at like, some homology search, look for some enzymes that were available out there for these top reaction, this conventional reaction, and screen for the other promiscuous reaction, the reaction that they usually don't do. They found one hit, this TP4H enzyme can actually convert this compound number 1 to have like this hydroxyl group. And that's pretty interesting because this could be a starting point for other, other aspects for our engineering, right? So for this paper, they take these enzymes that has promiscuous activity and do further engineering. The further engineering that they do is that they go into the structure and try to do some directed evolution through like design and like try to like screen for some that has activity. And they also found that like the stability of these enzymes are not great. So they take a step back and do the de novo protein design to make sure that the protein that they have were more stable as well. So you can see here that the pink bar were from the engineering of the native protein, while the blue the yellow bar came from the protein that were optimized, like initially optimized for the strong backbone first, and then do the same engineering to have these like single, double, or triple mutants. You can see that like the total turnover number, TTN, which refer to the activity of the enzymes, keep increasing by accumulated mutation of these enzymes. So that means that like we can improve the function of enzymes or like whatever enzyme that we're trying to make if we have like, good enough knowledge on how to do it. And we have like, proper setup to do these things. So this is an example of like how do we use these de novo design to boost enzyme activity and also like do some conventional enzymatic engineering or design systems. All right. Now getting back to our complicated, railway, metro, or whatever metabolic map we call, we can still see that like, what if we have the enzymes? How do we start? Because like we don't only want to make the compounds we want to make. We want to make a lot of them as well. We want to make sure that the process that we're making is, not cost prohibitive, like is appropriate for us to use this for chemical production compared to the cheaper way of like using the non-removable petrol chemical based system. So there are several ways to start from this system. First, let's think about, like what if our chemical reaction is going into a wrong direction? I'm not sure if the animation is going well, but you can see that these arrows were thinner. And there was another enzyme Z right here, enzyme Z prime, is taking this compound B and turn it into compound Z, which we don't want it. How can we like, get rid of these to make sure that we can still make our proper like ZDX product? Here is a question for you guys. What should we do? If we have enzyme Z prime, they keep pulling B away, what should we do? One, you can try to get rid of enzyme Z prime entirely, like just delete it out of like the system. Two, try to reduce the amount of enzyme Z prime to the minimum amount. Or C, find another route to make C that doesn't go through B. So I'll give you guys some time to try these out. I still keep seeing people putting their choices in. This is very interesting. The answers are very close to each other. I'm not sure if you can see the amount of answer going on or not, but we are pretty close. We are around like 20%, 30%, or 40% for each of the answer. So it seems like you guys are correct in different ways, because all of these three answers can be correct, but it depends on the situation. So this is kind of a trick question. I'm trying to trying to convince you that like there's no absolute correct question for any problem. There might be zero answers to a particular problem. For example, if this enzyme Z prime is not important for the cells, you can try to get rid of them, just delete them. In the terms of like strain engineering or metabolic engineering, we'll call them knockout. We just knock them out, get rid of that. But if that enzyme is actually essential for the bacteria to grow, then we won't be able to get rid of that enzyme completely or get rid of that gene completely. So we can try to reduce the amount of that enzyme to the bare minimum, just so that this bacteria can survive, but not enough to pull product B away from the system. However, even though that was a choice or that was like something we can plan on, it may not be possible, right? Because if the amount of enzyme C is required at the native level, and you cannot lower that no matter what, otherwise it will die. Let's say like the enzyme that was used for ATP production, for example. These are some enzymes that you can't intervene. Otherwise, you would just kill the cells. Like thinking about some core production of like the factory. If you get rid of like the main controller, then it will cease function right away. So sometimes the option 2 is also not an option. How do we do it? So one possible way, but a lot harder way, is to find another route to make C. For example, we can try to find a new way to turn A into C using a different enzymatic pathway. It may be like turn A into B prime first, and then B prime turn into C. This has been used a lot in metabolic engineering, because we usually start from a simple carbon source like glucose or like some other sugar, because that's readily available, right? And then if we don't want to go through all the complicated system that the bacteria use to grow, or like in their what we call central metabolism, we can bypass them and go through another route, so that you can make a lot of the compound that we want. All right. So we just asked the question about like, can we just add CRTL and CRTY at the same time? Yes, we should add both of them at the same time. But what if this enzyme C prime is a lot faster than the enzyme CRTY? Then we still have this problem, right? Because like the chemical reaction of B to Z, like the last letter Z, compared to the B to the orange C, will be competing with each other. And then like you may not have a lot of C, because like there goes a lot to that gray Z right there. So again, it depends on the situation, and we want to optimize these process as much as we could. All right. Yeah.
Can I ask a question? Do we have these scenarios existing in systems naturally? Of course, I know we are talking about metabolic engineering, but do we have scenarios where there are certain enzymes in particular pathways that will suppress the activity of other enzymes? Like now in this case, enzyme Z, the American way is Z, but the British is Z. Enzyme Z existing that will hinder the formation of C. Wouldn't that be counterproductive in metabolism, or this is just a situation that you just brought up?
Yeah.
Could it be that this occurs naturally, or you are using it to help us understand metabolism engineering.
Yeah, so this may not be exactly with this, lycopene pathway. There might be one, but like it may not be that strong. But I can provide you other example that it happens in this kind of situation. Like if you ignore, the CRTL, CRTY, there are some of these, example that we have to go through. We have to bypass, making compound A to compound C right away without going through B. And one example is what we call mevalonate pathway, when we're trying to make a lot of, terpenoid compounds, and like they have like a very like strong regulation system. If we go through a different route, we can actually bypass their regulatory system. And that in that case, we can make a lot of like the product we're looking for. That was also part of the artemisinin, the compound that we care about, and we have been mentioning a lot in our earlier, in our beginner course, and I will bring it up again at the end of this class. So thanks a lot for the question. Really, that's a very good question. This is real, and as a metabolic engineer, we see this all the time.
Thank you.
Yeah, let's move forward to other examples. So the other like abstract way of like explaining how to do metabolic engineering in the chemical engineering way is that like we can do something like push, pull, or block of these like chemical reaction pathway. How do we push it? Let's say that we have these like whole pipeline or workflow that like starting from here, like from the top here to go to the product. I'm so sorry about this. This is supposed to be the desired product. Let me change it right away. Now it's the desired product now. All right, so we have this desired product that we want to get, and we want to see like how can we improve the production of this desired product, the green one, but on the bottom right here. We can try to push it further, like like try to increase the rate of the chemical reaction that go through into this cycle, and also like go to this core desired product that we want. We can also do what we call pull. Pull means that like since these chemical reaction can can occur, sometimes they can occur in the equilibrium, they will make more products only if that product was consumed or converted into something else. So if you keep pulling on these later chemical reactions, you can actually increase the flux. That's the keyword here. The flux means like the rate of like the conversion of the initial compound to the other compound. If you keep pulling them, then you can increase the flux. You can think about this as like if you're on the road, or if you're like like trying to move water around in the different size of canals. So if you have like a bigger canal at the end, then you can actually pull water out much faster. The other important thing to do, which is similar to the knockout we were talking earlier, is to block undesired pathway. The red dot right here are something we don't want, and we want to block them from going sideways. And this is like what we call push-pull-block strategy, where we try to improve the production of our desired product. The other way that we do it is like by using whatever tools are able to fine-tune. So this is one example from my lab, to use like the CRISPR system to tune or fine-tune the expression level of different proteins or different enzymes. How do we do it? What you can see on the left-hand side here are different like expression level of the fluorescent protein. But if we translate those fluorescent protein into these metabolic genes, where we convert GTP, which is like the nucleic acid used for the DNA synthesis, convert them into this BH4, the compound that is relevant for the disease called phenylketonuria treatment. If we try to make a lot of these BH4 or beraptamine compounds, we may not be able to make a lot of them if we just like randomly throw the maximum expression of these three enzymes in. What we argue is that we have to tune each of these enzymes to the proper level. And by trying like 4 different conditions for each of the gene, we have up to 64 expression strategy that we can try on. Okay, I just saw some questions. So I'll go back to the initial question that come to my direct message. Can the optimum product of enzyme C be made to inhibit its continuous activity Yes, that's another strategy, but it will require a lot of protein engineering to work on that angle. Thanks a lot. That's a question from Ajit. Some of you may not see it because it's not asking publicly. There's another question from Sei. What are those things that we use to pull enzymes? So, yeah, you're right. We use enzyme to pull, the chemical reaction from one to another. And like the faster the enzyme is, the faster you can pull it, right? So you can also, again, think about the canal or the road. Like if there are some roads that where like the people can walk faster, that means that like the people on the earlier road can probably like walk faster to a certain extent as well. But at the end of the day, there'll be something called the rate limiting steps, where like if there's some roadblock that was too slow and like make people or make like the chemical flow very slowly, we have to fix that part and make sure that we do this push-pull strategy as appropriate. Does that answer your question properly, Say? Okay, I'll go for the other question then. How about the natural system whereby after a disruption, they tend to return to equilibrium when you push or pull? Yes. So like they try to return to equilibrium, right? So we have to keep pushing or pulling this equilibrium. It depends on the state of the cells or the state of the system that you're controlling. But most of the time, if you're trying to pull, we try to pull that like continuously, so that the pull of the equilibrium keep getting pulled. Or the same goes for the push. Like if you think about the chemical reaction, like the more starting material you have, the faster it can turn into the product. The less product you have, the more equilibrium will go to the product, right? So it's either like push to the starting material or pull from the product. Is that a question? Not really. Okay. Yeah, I hope those like, clarify the question that you have. Let's take a step back to our fine-tune scheme here. Again, we have these 3 genes. We have 4 expression level for each of the gene. So like if you do 4 times 4 times 4, you have 64 expression programs available here. All right. Here is what we tried. Like we tried to change this like expression level into high, medium, low, or like no activation at all. We can see that like, if you try to express everything all on, is not the top performers. Only if you like, tune something down, like in this case, is actually like the high expression of the first gene, the medium expression of the second gene, and the very weak expression of the third gene. Then you can get the highest like production of this like BH4 compound. What's the reason? The reason is that like when we go back to our literature, we found that like this enzyme, the third enzyme here, has a very fast kinetics. So you don't need a lot of those enzymes. And sometimes if you express too much of those enzymes, it actually pull the resources from the cells to express those enzymes that you don't really need a lot. So this means that like every time you're trying to do these like, optimization of the metabolic path or metabolic pathway, you have to be concerned that like it's not just like putting the genes in. You have to make sure that you do this push-pull strategy and also fine-tune the expression level of each step to make sure that they're doing, the right expression level and they're doing the right chemistry. All right. The next thing that we're going to talk about is this like in and out strategy, where like, you have some compounds that you're taking in. Let's assume that these like, 6-membered ring or 5-membered ring are sugar that we're taking from outside. And then there should be some ways that this sugar will get into the cells and then turn into the product that we want, and the product has to leave the cells, right? So like these are like, one concern that like if you're trying to make sure that the bacteria can effectively convert these sugar into the products, you have to make sure that they are capable of taking the sugar in, or whatever starting materials or the compound that you want to use as a starting material. They should be able to like take it in, and then it should be able to kick the product out as well. Because if they don't put if they don't like push or pump the product out of their cells, then that will make another kind of chemical equilibrium to stay still, and you might not be able to make a lot of products if it doesn't get outside of the cells. Sometimes you don't need these like, active transportation, because like some small molecules were really easy to get out of the cells. For example, if you work on ethanol is actually volatile, right? So like at some point, ethanol will just like float up and just get into the gas system right away. But some compounds, like especially something that has like a lot of charged molecules, they can't go through the cell membrane of bacteria, because these bacterial cell membrane or cell wall, they have like the lipid, right? The lipid doesn't let any charged molecule pass. So if you have any charged molecule that you're working on, you technically need some transporter to help them go through these like cell membrane or cell wall. All right. Therefore, like incorporation of these transporter proteins are usually pretty important if you're trying to do metabolic engineering, or you try to improve the metabolic engineering. You have a question? Oh, maybe that's just an unmute issue. Okay. I hope this makes sense. If you have any question, please feel free to like raise your hand or type down into the chat. I'll move into another strategy. So think about, like let's say we have limited resources. We mentioned a bit about limited resources, right? If we want to, make just enough protein, then that's one thing. The other angle of these like limited resources sharing is that like sometimes we don't want these bacteria to grow too much. Because to grow a lot of bacteria, to grow a lot of them in the population, they actually use a lot of these resources that were allocated initially. So sometimes we have to do some growth coupling or decoupling to make sure that like the resources that we provide were mainly used for the production of the desired products that we want, not making whatever bacterial cell wall or like, trying to make, energy sources for the bacteria themselves to make like, random proteins. We don't want that. So one angle of these strategies is to make sure that we allocated the resources for the growth and biomass as appropriately. Because if you grow too much of the biomass, you may have like little amount of resources left to make your product. However, if you make, if you like keep too little of these cells, then that means you have like too little of the enzyme as well. So it doesn't mean that like, you always have like higher product, because like you may end up with a lot of leftover resources as well if you have too little of the growth. So these kind of like growth coupling programs is one of the key things in synthetic biology that we can use to control how much cell density we want them to be, so that they can convert the starting materials to the product at the proper rate without like overly using, our useful resources for just making a lot of like their cell density. All right, that's another one. And the last one that I want to mention for our general strategy here is that like we have to choose a proper start point. Why is that? It's because like some bacteria are good at different things, right? So let's take a look at this like Kiattisewee. They're pretty good at eating sugar. They're okay tolerating the heat, but they might not be able to stay in the salt, in the high salt situation, or they might not be able to, eat lignin or carbon dioxide. Then in that case, they might not be able to, like be functional in the case where we want to use like some feedstocks that we want to get it in a cheap situation like lignin or carbon dioxide. I see the question here. How do you reduce the growth without affecting viability? And also, can you use quorum sensing for that growth-based work? Actually, Joy's question actually answered Abhijit's question already. We can use some of this quorum sensing to control the cell density. And for the viability, again, we have to make sure that we control on the cell density, not on the growth itself, right? So quorum sensing is one angle, one strategy to control these cell densities. Or like there are also other things. There are also other things that like, we can capture that and like program, make the cells turn on and turn off their expression profile as we want them to be. That's the magic of synthetic biology or engineering or like biological engineering that we have been learning so far. Thanks a lot for the questions. All right, so let's take a look. What if we take Kiattisewee that is better than A at taking lignin, but now I still like cannot tolerate the salt, but can stay in the environment that has like some organic compounds. This might be better now. And there might be some other organisms that were, pretty good at eating everything and also like tolerate to salt as well. So like, it depends on like how you're going to choose the chassis to do the engineering. It has to be clarified that like, most of the time, some of these new organisms doesn't have a lot of tools available. So I put some notable chassis on the left-hand side here. And like, these are some lists that like people in the field of synthetic biology are interested in. E. coli is so far the easiest organism to work with. And then like Pseudomonas putida or Bacillus subtilis might be like, another, handful of organisms that we can engineer them pretty easily. The like further along the line, like except yeast, Saccharomyces yeast, these are like not easy to engineer at all. So like, even though they're having like a very attractive, capability, they may not work as you want it to be. Like there are still other like, other angles of like choosing chassis, like growth or like, how do I say it? There are some other like tolerance to different condition as well. Like let's say some of them can't grow with oxygen around, or some of them has to grow with oxygen around, something like that. So those are some concerns for you to choose the chassis as well. So if you're thinking about your final project and you want to work on like some, different organism, you may have to choose the organism for your project properly. Or you may just like, try to screen for one organism that may work as you want it to be. And you can set your goal that you want this kind of organism and try to select through it. All right. So there'll be more discussion about these chassis on the microbes for sustainability discussion on lecture 7, the last lecture that we have. So like, stay tuned. I think Enrico will go deeper into like what kind of microbes we might be interested in. All right. This is like the end of the metabolic engineering lecture already, but I want to like give you an overview of what has been going on in our field. So these were figures that were took from like this review. You can technically like, use different system, including the machine learning or like the computational method to develop like a design and learn system to make sure that like you go through the system faster. And also like, miniaturize a lot of system and also like test a lot of things in parallel so that you can go through the build and test approaches much faster. Design, build, test, learn is something that like synthetic biologists or like metabolic engineer are going through all the time. We want to improve this like system further and further, so we have to go through this cycle all over and over. Okay, I have a question from Abhijit. Which out of 3 chassis would you prefer for metabolic engineering? One that uses a lot of types of substrates or less types of substrates? Very good question. So there are pros and cons for using the organism that use just a single substrate or use a lot of types of substrates. So most of the time, when the organism use only one substrate, they're very good at using it. But those who are like, jack of all trade, who can use anything, they're not that great at eating any particular, compounds. So they might grow slow if you give them suboptimal compounds to eat. And they will grow eventually, but they'll be slow, no matter like what compound you give them, something like that. But for like, let's say E. coli that likes to eat sugar or like amino acids a lot, if you give them these proper compounds, they will grow really fast. And if you want to make protein, maybe E. coli is the appropriate host to make a lot of that recombinant protein in a faster landscape. But if you want to make sure that like you can keep the cost low, and you want to focus on like the sustainability issue, sorry, you may want to focus on other bacteria, because E. coli like so far only like to eat sugar, right? Unless you engineer them to eat other things. But that's a big engineering scheme as well. All right, let's move forward to our other example. So there are some concerns or like some ideas about using machine learning to predict what we should be doing and then apply the automation or the self-driving angle. Let's say like think about like the self-driving car. What if we make a self-driving lab? You can take a look at this like opinion piece and they describe pretty well on like how do you use this automation system to push it further. The key point here is that like instead of like using a lot of resources to try a bunch of things, if you can use like machine learning or like AI to help like suggest you the proper thing to do, then you need less resources to work on this. At the same time, if you can automate it, then you may not need, you know, a very high skill human researcher to work on all these things, and you can also reduce the human error that occurred throughout the process. So that's kind of like, you know, another important point why we want to make like or elaborate this like AI or machine learning system or the automation and self-driving angle of these. All right. Again, before we adjourn, I want to remind you guys that like most of the thing we talk today in the metabolic engineering is based on the programming in the DNA level. So with the DNA, we have it work as a storage that like you can make protein at the end, right? Most of the time, you just put these like, you know, DNA program into the plasmid or the genomic DNA, and you can revisit like the lecture last week to see like what is the strength engineering in terms of DNA level that has been mentioned so far, just in case you want to, you know, use some of that for the planning of the final project. I want to like, you know, provide some example of like this transporter engineering. Sorry, that I talk about like in and out kind of in and out kind of strategy. So this is like one organism that were engineered to have not just the glucose importer, but also the cellulose, which is carbon-5 importer that has been you know, very abundant in cellulose or like hemicellulose or lignin. So by having this like, you know, another transporter protein, it can actually grow a lot faster in the mixture of these like two types of sugar, and that's, you know, that's very useful, and that turns out to be you know, a very important thing for these bacteria to grow. So this is one example, you know, because like I just want to showcase that like sometimes you don't really just engineer at the thing that is going on inside the cells, but in between, inside and outside the cells as well. This is our classic example of like how to make artemisinin. And the first question about like why do we have to bypass is because like, you know, the regular pathway of making this thing is pretty complicated. And by having both the MEP DXP pathway and the mevalonate pathway, have both of them on to make these DMAPP and IPP, can actually elaborate or you know, enhance the productivity of these two compounds a lot to make these terpenoid that eventually will turn into artemisinin production. If you only rely on just one pathway, or just the native productivity of these like you know, DMAPP or IPP, you will not get a lot of artemisinin. You have to push them from both ends and then keep pulling them from the far end here to make a lot of artemisinin. Again, it has to be declared here that like we can only make up to artemisinic acid, and this artemisinic acid will go through the chemical pathway to make this artemisinin compound. So far, like scientists have been trying to make artemisinin like, you know, all biosynthetically, but it has been very difficult to make this artemisinin like, you know, in the whole organism. So this is a lot cheaper to just stop at a high yield artemisinic acid and then like go to artemisinin by using and just a few steps of chemical synthesis to help. And the last thing, very last thing now, it just came out last week for the biosynthesis, like the complete biosynthesis of this vaccine adjuvant. So I think everyone appreciate vaccine, right? Because like the recent pandemic has like tell us a lot that like we need to make sure that our vaccine are working properly. So far, like to make this vaccine, you have to grow a big plant and grab the bark of them and keep extracting these barks. And it's been very hard to extract this like adjuvant QS21. So there has been some campaign to try to make these compounds first in plant, and it was done last year in tobacco. Like we can actually make these, like this is from UC Berkeley, from Kiesling Lab, which is like one of the pioneer of the metabolic engineering work, including the artemisinin work. So they can make this QS21 in tobacco last year, and now they've been trying so hard to put it into yeast, and they are successful just last week or a few weeks ago, and they can make this compound that look very complicated, Like you can see here that they have like 4 or 5 parts like connected to each other, and they can put into yeast, sorry, to make this compound. So like this is like, what I want to like elaborate here, that like as an inspiration, that if you are trying to make something, is not just making it, but to make sure that you can make a lot of them and also make them cost competitive. Otherwise, like people from the chemical industry plant, we just keep saying that like, hey, it doesn't matter. We just keep using, the petrol because it is like 1,000 times cheaper. We have to keep fighting. We have to do this hard and like difficult engineering to make sure that we can get these compounds and beat whatever chemical non-renewable system that is going on, so that in the future we have like a much better environment and sustainable system for us to move forward. All right, this is like the resources that we have. Like I also found that like there are 2 lectures from Chris Prather, another expert and pioneer in metabolic engineering, that you wanna maybe like take a look as well. You can get into the slide later and then see some of these like reviews that I put here, and also some examples of our work on some of the papers that we talk about today. You can see the list here. So that's the end, and I'll take any question if you have. Otherwise, like, we can adjourn early, or I can also like, answer any question about the group project if you have one as well. All right, I have a question from Dan asking, how do you handle byproducts in an engineered metabolic pathway by introducing byproduct removal pathway? This is a very good question. Thanks a lot, Dan. So like byproducts means like something that you don't really want, but you make them anyway. For example, if you think about the chemical reaction, when you're trying to make the acetone in our first slide, let's say, when we're trying to make acetone, we also have phenol together with it. This is what we sometimes call side product, byproducts, depending on like the definition. But it would be much better if the byproduct or the side products can be used in some ways, right? So it depends. If the byproduct can be catabolized. Catabolized is like a fancy word of eat. If the bacteria can eat that byproduct, that doesn't matter, right? They'll just take it back in, feed themselves with this like carbon source again, and keep making the desired product that we want. But if they cannot eat the byproduct, it depends on us whether we want to introduce a new pathway to remove those byproducts, or to eat those byproducts and turn them into some, some fuel, some like chemical starting materials for the cell to make our desired chemicals. Again, we have to keep in mind that like most of these metabolic engineering scheme or campaign, they're not always perfect because you have the biomass all the time, right? So those biomass sometimes can be used to feed, like, let's say like, if you like, scrape them up, they might be able to be used as like some food for cattle, for livestock, something like that. So there are some use for this byproduct or like side product of the metabolic engineering platform all the time. Or you can even like, feed this byproduct to another bacteria, another organism, if they can use it, so that they can turn that into some other valuable or value added compound later. Does that make sense? I hope that helps. All right. Is there any other question? I hope this is like, give you an overview of what metabolic engineering is. I try my best not to go too deep into what people really do in metabolic engineering, but like you can take a look at some of these like some of these attempts that has been done, especially on this slide right here. You can take a look at these lecture notes. If you have trouble getting some of these like materials, you can connect with us and maybe like we can help with that. So like there is a lot of fancy system to develop these design, build, test, learn approaches. I was working on one as well here at my lab here, and it's not easy. It's been difficult. But we are doing it for a reason, right? We want to make sure that our society doesn't rely on any fossil fuel, because we will run out of that one day, right? So again, this is probably like, a wrap up. If there's no other question, thanks a lot everyone for being such an active students for this for today. And again, I will post the homework on the syllabus. And I will send an email, a follow-up email about like our syllabus again. So please keep an eye on your email. I'll put onto Slack, and maybe Kato can put it onto the WhatsApp channel as well, so that you guys can reach out. All right. I think that's it for me. Kato, do you have anything to add?
Okay, thank you so much, Ice, for today's lecture. It has been really very wonderful. What I want to add is just something small. It's an announcement. I would like to announce that SynBio4All Africa also organizes what we call the SynBio4All Africa guest speaker events, whereby we normally invite key people in synthetic biology to come and talk about synthetic biology in a perspective of business, not only in a perspective of study. So this month, we normally host these events every month. So this month, we are privileged that we shall have one of the members from IGEM, and is the program coordinator at IGEM. Is called Yugo. So if at all you are interested in listening to his talk, he will be talking about the role of IGEM in building and supporting the synthetic biology ecosystem. If at all you're interested in listening to his talk, you can register yourself on a link which I have shared in the chat, and see you soon. That event will happen on 31st of May. That's Friday next week. Thank you so much, Ice, and back to you.
Perfect. Thanks a lot, Kato. Yeah. So even though we like, we have like our intermediate course going on right now, this event is separate from our intermediate course, and it will keep going even if like you're done with the class. Let's say you got certificate and everything is done. Please keep an eye on our like, activity or like our webinar here. I've been working with Kato to like, recruit more people to come in and like talk with us as well. At some point, maybe we might be able to invite some people from Africa who has been coming to the PhD here and explore more on how they can like, make the world better through the synthetic biology applications. So it will be very inspiring and also like very resourceful for all of you to attend. So please keep an eye on. All right. I think that's it for today. Thanks everyone again for attending and for your active participation. I really have fun like, going through the poll and like all the Q&A with all of you. And if you have any question, please don't hesitate to reach out to us either by email or on Slack or on WhatsApp. Maybe I will connect with Kato if I should hop onto WhatsApp as well to see if I can help anything. And also like finally, if you have trouble installing Slack, you can try to work on your browser. Your internet browser might be able to like let you get into Slack. So again, Slack is probably like the easiest and the best place to pop question in, because we have different rooms. But like, whatever channel you reach out to us, we'll be as responsive as we can. All right. I think I'll stop the recording right now. Thanks everyone again. Goodbye.